Awesome. Thank you again, everybody, for staying all the way to the last talk, and thank you so much to the committee for inviting me. I'm delighted to be here. It's my first time at CBIAS, and it's a fantastic meeting, and hopefully get to come back in future years. So I'm going to tell you two short stories about the work that we do at the imaging platform. The first is about how can we learn information from images more easily. And the second is about how can we learn more from the images that we already have, even information we didn't even know was already hiding in our images. And I want to start from here. I think this room, I don't have to convince anybody that images are data, right? We're all on board? Some rooms, that's not always the case. But we do have a problem in microscopy that we don't have in a lot of other fields, that people aren't used to thinking of images as data, because we have 400 years where they weren't, where they were pretty pictures. And now we're trying to tell people they're data, and they're like, no, wait a minute, I've seen these are pretty pictures. So we have a lot of baggage that we have to overcome as a community that someone who works in, say, sequencing doesn't really have to think about. But at the end of the day, images are arrays. And if we think about that, your standard microscope these days has a, somewhere between like 1 and 20 megapixel camera on it, that's 20 million pixels every time you take a picture. That's a lot of data. That's a lot of really rich, really dense data that you can, certainly we've seen a fantastic 2 days of all of the different things you can do with that data in terms of answering a biological question, but purely just from we have an array of numbers we can start doing fancy tricks with. This is an incredibly rich data source, but this paper is almost 10 years old now, but I'm not actually sure it would change that much if we did the same analysis today. Papers that called themselves high content microscopy mostly report 1 or 2 measurements of cells. So again, we've got 20 million pixels in each image, times channels, times z-planes, times fields of view, and we get one measurement? It doesn't really feel like enough. And so that was essentially why about 15 years ago, Ann Carpenter founded the imaging platform at the Broad. The idea of turning images into answers and getting some of that extra data out and getting more data. And so the first story I'm going to tell you is about work that's primarily happening in my lab, where we sort of turn images into numbers. And then the second half of the story is work that happens mostly in the Carpenter Singh lab, where they turn data into answers to questions. We make a lot of tools. Cell Profiler, you've probably heard of. Some of the rest of these you probably haven't, though I'm certainly happy to talk about all of them. And PicSimi, you're going to hear about right now. So the imaging platform, it's a really fun place because we do have all of these backgrounds in one sort of we're a self-contained image analysis unit. There's no microscopy core at the Broad. We're not part of the IT team or even the software platform. We are our own sort of funny little image analysis group. And we make new software to make bioimage analysis easier and more powerful. We work on training and how to come up with new ways to train biologists into bioimage analysts, as well as new assays to make drug discovery and biological discovery faster and cheaper. We also do collaborate with folks everywhere, globally, nonprofits, academics, companies, and so we're always happy to work with people. If you have a problem that you think Cell Profiler is the right answer to, we're always happy to have new collaborators. So I don't know if you've heard of this deep learning thing. I think it's got legs. I think we're doing this. And there's a reason why people are so excited about deep learning, and you've heard about it so much, because there is a lot of things that it can help us do in microscopy. Some of this is stuff from the imaging platform, some of this is not, some of this is just sort of very cool work that's happened elsewhere. But all of these are examples of things that you can do putting deep learning onto pictures. You can predict assays, you can get rid of batch effect correction, all kinds of fantastic things. But so here's the problem. This is a survey that we've now run a couple of times. Most recently, we were lucky enough to get some great folks who are in this room to help us analyze it in terms of asking people who analyze images, whether they've ever analyzed one image or millions, about what they're interested in, what kind of analysis they want to do, what's easy, what's hard. If you want to know more, this just came out in the RMS, the Data Analysis special edition, so thank you guys for having us there. But if we ask people to describe their work in terms of how much are they doing imaging, making samples, actually operating a microscope versus doing analysis, and then we also ask them about their comfort in developing new computational skills. We have a separate question where we ask them, what are your skills already? But just how comfortable are you learning something new on the computer? You might not be surprised to hear that the more of your time you spend on a computer at work, the more comfortable you are developing new computational skills. That's not really surprising, but it does mean that the expert on what your phenotype looks like often might be in some of these very low computational comfort categories. And the person training your DL models, who again, there's amazing people in this room who are biologists who train deep learning models, so I'm painting with a very broad brush here. These might be totally different people, and they might not even have a good way to talk to one another. Again, I think this is the room where that isn't true, but I don't think, unfortunately, the rest of the world looks like this exact room. So, assuming that we all don't want to work, 168 hours a week solving image analysis problems for people, which I'm going to go ahead and assume, what can we do? So we're really excited about the power of web tools. As somebody who, spends a lot of time making and maintaining Cell Profiler, which is not a web tool, is a desktop software. Desktop pieces of software have some advantages, but they also have some disadvantages. And especially if you want to reach out to these very computationally uncomfortable users, getting rid of installation is a huge, huge help. When in this paper that I pulled this figure from, we actually looked, and in the image.sc forum, if we went back about 6 months of posts, 1 in 10 posts has install in the first, the initial post from the user. 1 in 5, the word install comes up eventually. So this is not a trivial problem. This is actually a huge problem. And I added this slide after yesterday. So this is an example of how we can use containerization to get web tools to people in a no-code way that's easier. In 1 minute here, I'm going to search for a Docker container, download the Docker container, and then start a web app where I can do, in this case, it's Atsu thresholding, but what could be in this container is whatever. This could be hosting your deep learning model. And the code for this is online. This is just a sort of little demo app. Callum Termiskoya, who is a great postdoc with me and is now a great postdoc with Virginie Ullman, put together the original version of this. And this is all regular speed. This is a Docker container, but it is creating this very nice, simple web interface. If you've ever used Hugging Face before, this is the same library they use. We can make it easy to use web tools to very easily deploy bioimage analysis tools. And so this is great. Again, this video is 1 minute long, and it goes from searching for the thing to getting out segmentations. But the person has to go find the thing. So already this is too hard, and they have to have Docker Desktop installed, which is, they go to a website, they click, give me this, but that's still too hard. So that's the idea behind Piximi. So Piximi, our tagline for this is images to discovery. And the idea is, what if we could serve deep learning models to people in their browser, not client server, not saying, hey, you upload your images, send us to our server, we'll do some processing and send it back to you. What if this could all happen and all your deep learning could be trained and run in your browser on any device that you have right now? And so you can go to piximi.app on your phone or your laptop right now. It's going to look like this. We have images and we have objects that you can look at and view. And we have an annotator because we know that people are going to want to use this to train segmentation models. Right now, trainable segmentation is coming. Right now, you can only run pre-trained segmentation models. But the idea being that we're going to be supporting fine-tuning of deep learning models in the browser. We need to have some place where people can actually fine-tune the outputs of models. And so our annotation functionality has a lot of the fun toys that you might like, bounding boxes and brushes and stuff. But we also have superpixels. And this one's my favorite. This is the color annotation one, where you just sort of click and drag, and then you hit enter, and then all of this is nice with hotkeys. And the idea being that you have in a web browser where you never installed anything, you just went to the website on whatever device you happen to have, you can now start annotating your data really nicely. And then if you don't want to annotate it, if you want to use a pre-trained segmentation model, and again, soon to actually do segmentation, you can just do that. So this is real time, this movie. And so I've said, hey, I have this image. So in this case, I want to run it in CellPose. So you can connect this to a server if you want. So CellPose, we actually connect to the bioengine, but Piximi warns you that it's going to do it. But if it hasn't told you, hey, I'm going to a server, all of this is happening locally client side. And so here I've now accessed CellPose. but the user has never seen anything that looks even remotely like code. When the segmentation training functionality is built out, which we hope will be soon, it should look pretty much like the classification thing. We also have object detection. So those of you who missed ELMI this past year, this is what ELMI looked like, and you can see that it finds some pictures on the beach. Here's classification. And so you can see here we have hyperparameters, but all these hyperparameters are hidden from the user. And so here I am training in the browser, again, not client server, in real time, a model that is trying to classify 3 classes of cells: cytoplasmic GFP, no GFP, and nuclear GFP. And I can watch my training history. All of those hyperparameters are there and are tunable. And if I know what those are, I can go in and I can actually do something with them, but I don't have to. And we can make it easier for users to sort of get into deep learning slowly by giving it to them in a way that is no install, that is very easy to use, and where then they can go through and learn more about the hyperparameters as they sort of turn knobs to see what works. And this does work on any device that can access the internet. So this is on my phone. I will have you know, I did not show this to people at the bar last night on my phone. I promised my software engineers I wouldn't, because they said it's not cool to do that. So I didn't, but I could have. So we're really excited about this for the purpose of, we all know that there's a lot of amazing fancy clusters are hanging around in our community, but also there's a lot of 12-year-old PCs that somebody is keeping together on their last legs. And certainly that's even in the global north. When we get into the global south, the access to compute is going to be even worse. And so by making tools that make it easy for people to run models and train models, and I apparently took out this slide just for time, but you can then save the project file and then give it to somebody else so that they can see your annotations and see your training, and all of the models use standard formats, COCO format, things like that. So we're excited about the idea of web tools, and this web tool in particular, in terms of taking all of the amazing kinds of deep learning that people in this room are inventing and making it easy. So now I'm going to switch gears a bit, and I'm going to tell you a little bit about the other half of my story, which is about, great, so that's all information that we knew was in the images, we knew what we were looking for, and we went and we found it. What about my unknown unknowns? How can I get information from my images I didn't know was necessarily going to be there? So again, in this room is probably one of the few places I don't have to explain the idea that, images are data, and we can take cells and we can turn them into measurements, and essentially this is a latent space. We have a feature vector now that describes a cell, and it's very straightforward to take cells and turn them into a feature vector and learn things. But certainly 10 years ago, this was not the standard approach. And the idea that you could take these feature vectors and actually learn interesting things and learn novel things was not something that was initially completely straightforward. So about 15 years ago is when, at the Broad, Ann Carpenter and Stuart Schreiber started putting together what we now call the cell painting assay. The idea being, how many dyes can we, as cheaply as possible, and those of you who do microscopy may recognize some of these things as things that are in the back of your freezer, to just get as much information of a cell as we can so that we can turn it into a high-dimensional feature vector and then just do unsupervised clustering and sort of see what kinds of things fall out. And so this is designed to work on any sort of standard high-content microscope. Counting replicates, it's about $1 a sample once you're running it at scale. So this is very inexpensive to run, and it can be run on any 4-color microscope. And so I'm going to sort of skip ahead to just telling you, I did not believe that this worked when I joined the lab, because I was like, there's no way. You should be targeting the specific thing you want to find out about. But it turns out that this works. So if you're not a RAF-RAS pathway biologist, that's fine, neither am I. But what you can hopefully see is that replicates of different RAF-RAS pathway genes, the replicates all cluster with one another in cell painting feature space, and that these are mutants of these genes, which are the next cluster over. So not only can in cell painting feature space, we can tell that the replicates go with each other, that the things in a pathway belong together, but that cancer-causing mutants in these genes are similar but not quite the same. So there's actually a tremendous amount of subtlety you can get once you sort of get into this cell painting feature space. And so Mohammed Roban, who was then a postdoc and now has his own lab, spent a lot of time looking for pathway-pathway interactions, found a lot of stuff that was known, which when you're validating an assay is always a nice thing. but found at least one that was not well literature supported between NF-kappa B and Hippo. And those looked anti-correlated in cell painting feature space. And so since we're a totally computational group, we had to find some friends who would make us a reporter assay, and I'm telling you the story, so you may not be surprised to hear that yes, when you look at a Hippo reporter assay, NF-kappa B, which is opposite in cell painting feature space, overexpression turns the reporter off. So this is novel pathway interaction biology from organelle dyes at $1 a sample. So I'm going really fast through this. May have time for lots of questions and or extra slides. So we're now starting to be able to do really fun things with this. And again, in this room, people probably are not so skeptical that latent spaces and feature vectors have a ton of information, but this is one of my favorite ones that was just accepted and is coming out this year. We worked with a bunch of other folks at Broad to take iPSCs from a variety of donors, and they did whole genome sequencing and SNP calling, as well as we did cell painting in parallel. And what we wanted to say was, certainly one of the first question we get whenever we hand over cell painting clustering results to a biologist is, well, what's the feature? And why is the feature? And what is actually driving this difference? And we can usually point them at a feature name, but we don't always understand what it means. But in this case, we wanted to say at an individual SNP level, are there features that we can pull from cell painting that tell us about biology? Now we have 297 donors and thousands of SNPs and 3,500 cell painting features. So the curse of dimensionality bit us like whoa on this. So just getting anything to be statistically significant enough after correction was very difficult. But we were able to pull out 3 that we were pretty confident in. And there's a lot more interesting stuff in here that just, before feature correction didn't quite pull out. And so we found 3 different features that actually correlate really highly with a particular SNP of, all of these were genes or SNPs of unknown variants. And so, this could be an accident though, even though we've done a tremendous amount of correction for that. And so Matt Tegmeyer, who just left Broad to start his own lab, actually went and CRISPR'd all of these 3 changes in. And for all 3 of these changes, you actually get the results that you expect. Now, what I think is one of the fun things about this is, so of these 3 features, and again, with the advantage here of using conventional features is 2 of these 3 are not actually human interpretable. Human eyes literally cannot see them. But this middle one, PRLR, is in a feature that humans can see. But 2 of the 3 wouldn't have been and never would have been picked up by somebody looking at the images. And even this one where if you know what you're looking for, and in this case what you're looking for is that around the nucleus, the mitochondria are more symmetrically distributed than otherwise. If you know that's what to look for, you can blind these and unscramble them correctly. We tried it ourselves. But if you didn't know that's what you were going to be looking for, these look pretty similar. And so why is this nuclear hormone receptor causing changes in mitochondrial symmetry? No idea whatsoever. Somebody should really study that. But what we're now able to do is have a biomarker, and we can start now making lists of biomarkers that connect to individual SNPs. And so there's a lot that you can potentially do here, again, especially given this assay is cheap and easy to scale. And because these assays generate a lot of data, we're now starting to be able to do really fun things where we can, for example, take chemical structures and cell painting profiles and gene expression profiles and put them all in a model and shake. And then say, can we predict other assays? So cell painting is super easy to scale as long as you have the ability to do 96 well or higher plate microscopy. Some of these biochemical assays, for example, are not easy to scale, or they're bacterial or yeast or fungal assays, and so they're not going to be as nice for imaging, certainly, as sort of nice, big eukaryotic, mammalian cells. And it turns out that, 50% of the time it works 100% of the time in terms of training assay prediction from one modality to another. But if you want to try to train assay prediction from something where you have a big database of cell painting features, it turns out having a big database of cell painting features versus, say, chemical structures is actually going to allow you to predict more kinds of assays. And again, some of these are, yeast assays or bacterial assays or biochemical assays, you know, why exactly is one a predictor of the other? It's, you know, we certainly didn't look at that for everything, but it says that, okay, these really rich feature sets, if we just think of them as feature sets and not even as images, there's a lot of data we can start using in really fun ways. So now you're thinking I'm taunting you because I'm like, hey, look, we have all this data. Look at all the cool models we can make. And you're now thinking this doesn't affect you. But it does, because we've actually put all of these cell painting assays, and in fact, basically all of the cell painting we've ever done at the imaging platform, online in what we call the cell painting gallery. So Aaron Weisbart in my lab, as well as Shantanu Singh, and with a big shout out to Aaron Chu of AWS, who has somehow convinced AWS to pay a couple $100,000 a year to store all of our data for us. We're very grateful. And to give it away for free. This data is CC0. Any one of you can go take this data and do whatever you want with it. We hope that you'll cite us. We hope that you'll tell us what you did, but you can access this no matter where you work or who you are. So just to give you a little bit of a flavor of what is in there, the jump cell painting data set. We released this about a year ago now. So here we had 10 different pharma companies, because Ann Carpenter is a literal actual wizard who got 10 different pharma companies to all work together and sign one legal agreement. As well as some ORF overexpression data and some CRISPR knockdown data. So we have 117,000 compounds, all of which were imaged in 5 different physical locations at 5 different pharma companies. The customs was fun, let me tell you. As well as these 12,000 overexpressed genes and 8,000 CRISPR genes. And all of that data is cell painted with the same batch of reagent, and that data is all online. So again, this was a huge effort over the course of 4 years from a tremendous number of people, and we never could have been able to do this without academics and industry partners working together, which is, again, what I think is so great about this community, that these people are willing to work together. And the other sort of data set that I'll point out to you that's there, there's a lot more that I'm not telling you, but just a fun thing that finally came out this year after 6 years of work. What if we could do cell painting but pooled? So if you want to do a whole genome screen for CRISPR knockdown with cell painting, that would take about 200 384 well plates. Or you could do pooled screening, do in situ sequencing of the CRISPR guides, and the folks in the Blaney and Neal labs at the Broad developed this version of cell painting that was actually de-stainable, so that we can do cell painting, measure all the cell painting features, and then do in situ sequencing. So we have up there 3 whole genome pooled CRISPR screens, where each one is now 10 plates instead of 200 plates of data. And this preprint just came out again a couple months ago after 6 years of work, showing that you can discover all sorts of fun things using this pooled cell painting data. And again, you can see things that should cluster together, do cluster together, and vice versa. So I hope you guys make something cool with all that. Hopefully at this point you're A, awake, but B, thinking, oh gosh, this is really cool, I want to learn how to do this. For those of you who are firmly on one side or the other of the making images or making data, and you want to learn the other side, and or if you're just somebody who teaches this stuff a lot, one really fun thing that we did this year was create bioimagingguide.org. This was along with folks in the BINA Image Informatics working group. So this is the idea here is we have a Jupyter book of what are all of the things you need to learn to do a quantitative light microscopy experiment. We're not going to teach you all of the things, but we're going to at least list them and then point you at resources for all of them. And we were very inspired by Pete's Jupyter book, which if you've never checked his out, you absolutely should. It's amazing, but we get fun things for free in Jupyter book, like the ability to do drop downs and notes in the margins and things like that, and ways to just make this easy to learn and not scary. We can hide things inside drop downs and just make this so that any user who wants to can get at it. And when I say any, I really do mean any. We're now trying to make this multilingual, and we have our first language actually now that's completed, which is Czech. So if you go to cs.bioimagingguide.org, you can see this entire book in Czech. And we are working on a number of other languages, always happy for more volunteers. So if there's a language that you speak that you don't see here and you think it would be helpful to have an introductory imaging and image analysis guide, come talk to me, happy to help get you set up. There's initial automatic translation, and then the expert just has to go in and correct what's wrong. You've heard about forum.image.sc a bunch, so I won't belabor it, but yes, this is where all of us hang out, so please come here and ask us questions. And the other thing is just, again, we love working with groups from all different parts of the scientific community and making better tools for them, so please email me, come by the Broad if you're in Boston. And speaking of Boston, even if you're not in Boston, we're having a hybrid deep learning for bioimage analysis symposium, shameless self-plug, a week from Friday. You can sign up for free to listen to some cool talks from some cool people and just sort of try to bring the energy of sort of this community and this group of people over to the US where there is much less of this stuff happening. And so with that, this was the work of a tremendous number of great people, most of whom were not me personally, but the amazing people in my lab and the Carpenter Singh lab, and some of the stuff we make that we hope is useful to you, some of the people who paid for it, and thank you folks for your attention.
Okay, thank you. There is time for 3 questions at least. I see one is just being presented.
There is one online ask about Piximi, and the question is, are there any limitations in terms of the size of the images that can be processed?
That's a great question. So for right now, while the processing is happening locally, it's going to be very determined by your local hard drive. I'm going to be able to load in images that are much bigger on my MacBook than I am on my phone. We are working on remote backends. Again, I showed you that we can connect to the BioEngine, and the more we can do that, the better we'll be able to support larger and larger images. But right now, it's going to be incredibly hardware dependent.
Hi, Beth. Great talk. I'm guilty of doing many high throughput screens that are not high content. And actually, maybe you said it, but are there like scripts in any language like R or Python around for the downstream stuff, like after I get the SAR profiler tables?
Let me introduce you to PyCytominer, which is a lovely library for doing exactly that. And the preprint was supposed to be out last week, and I need to email Greg and see why it isn't. But yes, we have notebooks and code bases, and there's actually the image-based profiling handbook, which I can send you a link to, which actually details exactly all of the steps you should do downstream. So
thank you. Cool. That's
yeah.
Good.
Hi. Thank you for a lovely talk. My question is, if you could add one more channel to cell painting, what would it be, and how would it improve things? As on a more general point, I mean, there are obviously commercialized versions of, you said it was originally a very kind of cheap way of doing it, and what do you think the value is in the commercial ones? Thank you.
Yeah, no, that's a great question. So we've made a ton of variants of this assay at this point, where we've dropped out one stain and put in another, or we've had people add long stroke shift dyes. And even what we've seen with sort of doing feature dropout experiments with the regular channels is there's a tremendous amount of mutual information across channels, which is maybe, again, not that surprising. And so if you don't care so much about then being able to trace it back to individual features like I showed you, where did I show you that? Here. Then, you really kind of just need bright field and 1 or 2 fluorescent channels, and then your ability to do most of the downstream tasks is about the same. So add the channel that has the biology you care about, and then you can get in the same assay something that has specificity for your favorite biology and the ability to connect to these sort of large scale things. But in terms of just sort of information of replicates finding each other, it seems like bright field plus 1 or 2 fluorescent is sort of enough.